Hello, my name's Leanne Mooney and today I'm going to talk to you about human immunity. Types of transmissions. There are direct contact, indirect contact, insect bites and food contamination. Direct contact is the easiest way to catch a disease or infection. This is by touching someone or something with the disease or infection, for instance humans or animals. Indirect contact. This can occur when infectious people touch an object that you later come into contact with. Insect bites. This is a very common cause for diseases such as malaria, really prominent in countries like Africa, but there are many more diseases that can be caught from insects. Food contamination is one that a lot of people will come into contact with, and this is where you get food poisoning, and it is where the food or liquid enters your body, therefore contaminating it. The first line of defence. Both first and second line of defences are non-specific. They do not discriminate against different enemies. This means that they respond in the same way against every single attack. Below are some information pictures for you to look at. So one line of defence, what we use is through the skin. So the epidermis is the outer layer, and that provides initial mechanical and waterproof barriers between ourselves and the invaders trying to get into our bodies. The dermis. Now the dermis is the middle layer of skin. It contains many specialised structures such as blood vessels, nerve endings and hair follicles. The subcutaneous layer. This is the deepest layer of the skin. It's mostly made up of fat, connective tissues, larger blood vessels and nerves. It also works as a shock absorber against the body to protect it. The mucous membrane. Mucous membranes help your body remain in its healthy status by allowing nutrients through, whilst defending against infectious agents. A fantastic example of this is in your nasal cavity. They are lined with mucous membranes. While the surface is covered with cells and produces mucus and fine hairs called vibratory cilia, below the surface is a network of capillary blood vessels. The nasal cavity is the respiratory system's first line of defence. When we breathe, we can't differentiate between microorganisms, so our nasal cavity helps by filtering out pathogens so that they do not enter the body. The nose heats and humidifies more than 12,000 litres of air every single day. When we sneeze or blow our nose, we remove the mucus and any pathogens or particles that have been caught. Both the skin and mucous membranes release chemical secretions which restrict the growth of microbes on the surface. If pathogens cannot enter the body, they cannot disrupt the normal physiological functions or cause disease. The second line of defence. Inflammation. This is an increased blood flow to the site of the injury, to increase white blood cell count at the site of infection. Fever. This is brought on by a sudden rush of blood. It raises the core temperature and encourages the body to use an antibiotic effect and will continue to fight the infection until it is healed. Phagocytes. These are large white blood cells whose job is to consume all foreign bodies. Complement proteins, such as cytokines, are small proteins which help remove foreign particles. On the next slide, there will be photos to help you with this. Here below is an image to show you how the second line of defence works. The third line of defence. What are pathogens and antigens? Well, pathogens are microbes that can cause an immune response. Could be bacteria or viruses, also protozoans and fungi. Antigens are the part of the pathogen that tells your body it is under attack. The immune response. Acquired immunity is not present at birth. It is a learned response. It is formed by your life experience, the immunity you gain over your lifetime. As we live our lives, we pick up antigens from our environment, and our immune system learns from these and creates antibodies specifically to fight them. This learned response will help protect us for our entire lifetime, knowing exactly what to give you and when you need it. For example, when an antigen enters the body, the acquired immune system detects the antigen and works out a tailored attack for that specific antigen. Our immune system also learns, adapts, and remembers. It's extremely clever. Immune system cells. Monocytes. Monocytes are the largest of the different types of white blood cells within the body. They are made up in the bone marrow and take up about 5 to 10% of the white blood cells that are circulating around your body. Eosinophils. These are important cells in relation to allergies and worm infestations. They make up about 0.5 to 1% of the white blood cells in your body. However, it may be a higher percentage if you are having an allergic reaction. They work by releasing toxins to kill pathogens, mainly parasites and worms. Neutrophils. Neutrophils are the most common type of white blood cell. Between 40 to 60% of your white blood cells are these. They are a medium-sized cell with an irregular nuclei. Neutrophils are the first cells on the site of an infection. They act by eating the invaders. This is a process known as phagocytosis. Lymphocytes. These are small, round cells which have a large nucleus. They are made up in the bone marrow and then to be found in the lymphatic system. They make up between 18 and 42% of the white blood cells in your body. They are either B cells or T cells, and they play a major part in the immune memory. Basophils are the rarest of the white blood cells. They can be found in many tissues throughout the body. Immunoglobulins G, A, M, E, and D. The typical immunoglobulin is made up of 4 chains. They are arranged in Y-shaped configurations and generally consist of 2 heavy and 2 light chains with variation in polypeptide formations, allowing each immunoglobulin class to function in a different way during different immunoresponses. G is the most common immunoglobulin. It is able to stimulate a direct attack on anything that it recognises to be dangerous. G is the only kind of antibody that is able to transfer across from the placenta to the fetus, which provides some form of protection from pathogens it can't have encountered yet. A plays an important role in the cellular defense of mucous membrane to the body, for example, inside the mouth. M is comprised of 5 monomer immunoglobulins to create the pentamer in the first immunoglobulin to appear in the first sign of the antigen. E responds to the parasitic infections and allergic reactions, and D can act as an antigen receptor for B cells. Self-recognition and tolerance. The 2 fundamental factors which allow the immune system to tell the differences between itself and the foreign bodies are 2 sets of cellular surface proteins known as major histocompatibility complexes, MHC 1 and 2, along with the human leukocyte antigens known as HLA. When the immune system encounters an antigen, if the MHC cell is on the surface, it will not attack it or signal it because it is recognized as self. However, if the immune system encounters a new antigen it has not seen before, it will be recognized as non-self and the adaptive immune system will respond appropriately. The human immune system does not have a consistent level of response to foreign antigens, so the level of response varies significantly depending on the similarities between the new antigen and itself. This means that the new antigen is similar. There may be a weak response to whereas if the antigen is completely different, there will be a stronger response. Failure of self-tolerance. Hypersensitivity is an abnormal immune reaction caused by repeated exposure to the same antigen. Hypersensitivity has been grouped into 4 categories: 1, 2, 3, and 4. Type 1 is an immediate reaction. This is mediated by the IgE antibodies. An example of this is an allergic reaction. Type 2. An example of a type 2 reaction would be a reaction to drugs, medication, or even a blood transfusion. Type 3. Immune complex. This can be caused by inflammatory response, and an example of this would be rheumatoid arthritis. Type 4. Cell-mediated. An example of this is type 1 diabetes. Autoimmune or autoimmune disorders. There are many types of autoimmune disorders. The 2 types I will be describing are rheumatoid arthritis and type 1 diabetes. Rheumatoid arthritis is categorized as a type 3 hypersensitivity. It's a long-term autoimmunity disorder that primarily affects the joints. Common symptoms are warm, swollen, painful joints. They can be fluctuations where symptoms sometimes worsen. These are called flare-ups and then can lessen during remission. Type 1 diabetes comes under hypersensitivity type 4. It is also a long-term autoimmunity disorder. It is where your immune system can attack the insulin-producing cells in your body. Common symptoms of type 1 diabetes are excessive thirst, frequent urination, blurred vision, and excessive weight loss. Autoimmune conditions also have a tendency to be co-occurring. When you get one, you can easily develop more, although the reason that the co-occurring autoimmune disorders are so common isn't exactly known yet. Sometimes the body can reject transplanted organs. There are 3 types of rejection. 1. Hyperaccurate. This occurs a few minutes after the transplant when the antigens are completely unattached. Acute. This can occur from any time in the first 3 months. All organ recipients have some amount of acute rejection. Chronic. This can take place over many years. This is where the body's immune response slowly damages the new organ. We have 2 methods of acquiring active immunity, naturally and artificially. Naturally, these are the antibodies that have been self-developed by the body as a reaction to a disease or infection, whereas artificially comes from the vaccinations, where the body is intentionally exposed to the latest version of an infection and develops antibodies ahead of time. Both types of active immunity develop over time, but will last throughout your lifetime. On the other hand, we have passive immunity. This again is broken down into natural and artificial. Natural passive immunity is antibodies received through the placenta or through mother's breast milk, which does as well for around the first 6 months of life. Artificially passive are antibodies received from medicines. Common example of these are found in antitoxins or antivenoms, which neutralize toxins or venoms that we may encounter. Unlike active immunity, passive immunity is quick working, but they don't last very long. Immunological memory. Immunological memory is the ability of the immune system to respond to a pathogen that has been previously encountered. The primary response is when the pathogen is encountered for the first time. It takes time for the B cells to identify and find the appropriate antibody in order to start tackling it. However, this allows time for the infection to take hold and can sometimes cause more symptoms. Memory cells are released upon encountering something new in order to remember the correct antibody on a second response. This will take the immune system much less time to produce a high number of antibodies before symptoms are exhibited. Second response is the same pathogen encountered for a second time. The memory cells recognize the pathogen and produce high-quality antibodies which fight the infection before the symptoms arise. So what is a vaccination? A live vaccination is comprised of viruses and bacteria that have been weakened in a lab. When live vaccines are used, a relatively small dose of the virus or bacteria is given. This replicates to the body to create enough of the bacteria to stimulate an immune response to cause our antibodies to go through a sort of simulation before we run into an active cause of that disease. Inactive vaccines. Inactive vaccines tend to require multiple doses. The first dose do not give immunity, but only primes the second dose. Herd immunity. Herd immunity can effectively stop the spread of disease. When a high proportion of the population is vaccinated, the chance of people catching it is reduced. As communities become larger and travel is easier than ever before, the chances of diseases spreading are higher than ever. This has been experienced with COVID-19. More people mixing from things like shopping, holidays, works meant that people infected would spread to hundreds of people every single day. However, the reproduction number varies greatly on the disease. Microbes have all different characteristics which can affect the human rate of infection. For example, COVID-19 appears to have spread quicker than influenza and from person to person. This was evident in different groups. Vaccinations help. Vaccinations help against serious diseases. Viral diseases cannot be treated with antibiotics and can only be controlled by immunization. They help save the healthcare system lots of money in the long run, as prevention is much better than cure. The effectiveness of vaccines was evident within the measles vaccination programme. According to the vaccine site, after the vaccination programme started against measles, the deaths and notifications both fell by around 65%. Then a further 87% following the MMR triple vaccine introduction in 1988. Vaccination uptake. Unfortunately, over recent years, there have been many scares over vaccinations, which in turn creates a certain amount of misinformation and amplified stories. This makes it difficult for people to find out the true facts about vaccines. This was extremely evident within the COVID-19 outbreak. Lots of people refused to get the vaccinations due to it thinking it was not tested properly, but all vaccinations are regulated and tested before given to humans. This is the page where all the references are for the information that I have got, so that you know it is accurate and factual. Thank you for listening, and I hope you've enjoyed my presentation.